Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR PYQs of December 2023 shift 1 or morning shift. I will be discussing biochemistry, cell biology, applied biology, methodology, DNA replication, protein synthesis and genetics. If you are new to my channel, please do like, share and subscribe. Coming to the first question. The central rod domain of keratin protein is 300 amino acids in length. What is the approximate length in Armstrong of the rod domain if the peptide consists of distorted alpha helix, true alpha helix, antiparallel beta sheet? So keratin protein, it is 300 amino acids in length. And what is the approximate length of the rod domain if the peptide consists of true alpha helix? We will see that first. So a single turn in an alpha helix typically has 3.6 amino acid and extend about 5.4 Armstrong. So total number of turns to accommodate 300 amino acid residues will be 300 divided by 3.6, that is 83.33. Hence the length will be 83.33 into 5.4 Armstrong. That will be approximately 450 Armstrong. So 3 and 4 are not the right answers. We will see about 1 and 2. Then in antiparallel beta sheet, the amino acid extend approximately 3.5 Armstrong per residue. Thus for 300 amino acid length, 300 into 3.5 Armstrong per amino acid, that will be approximately 1050 Armstrong, that is equal to 1041 Armstrong in options. So that is seen in option 1. Now coming to distorted alpha helix. Distorted alpha helix, the length will be less than 450 Armstrong because it is distorted. So it should be 425. So option A or option 1 is the correct answer. Then which one of the following nucleic acids with the same concentration in water will form stable stem loop structure upon annealing by heating and flash cooling on ice? So you have to see out of these which one will form complementary base pairing with each other. So G doesn't pair with G, so it cannot, in first case, so it cannot form G. G doesn't pair with G, so it cannot form stem loop structure. See the second one, C is forming, will pair with G, again C will pair with G, again G will pair with C, A, U, U, and the rest of it will be the loop formation because it will not pair with each other. That is as shown in the picture here. So answer here is option 2. Whereas others, if you see, it is A, U, okay, they will pair, but U, U, they will not pair. A, G will not pair. So 3 and 4 also cannot form stem loop structure. So the answer here is option 2. Then next one. The amino acid sequence of tetrapeptides PQR is shown below. As calculate the net charge on the above tetrapeptides at pH 7. If the mixture of the above tetrapeptide is separated on a cation exchange column at pH 7, which tetrapeptide will elute first? Choose the correct answer given below. So P, this is the first tetrapeptide we are going to see. Aspartic acid, glycine, aspartic acid, serine. Aspartic acid, glycine, aspartic acid, serine. So this tetrapeptide, it is having an N-terminal amino group and a C-terminal carboxy group. Now aspartic acid also has a side chain carboxylic group. Glycine doesn't have any side chains. Again aspartic acid has got a side chain carboxylic group. Serine also doesn't have side chain, ionizable side chains. So if you see the pK of terminal amino group, it is 8 and terminal carboxyl group, it is 3. Whereas those of side chain of aspartic acid, pK value is 3.6. Now you should know one thing, that if pH is greater than pKa, if pH is greater than pKa, then deprotonation occurs and you will get a negative charge. If pH is less than pKa, protonation will happen and you will get positive charge. So if you see the terminal amino acid pKa is 8, pH is less than pKa, so it will be positive charge. If pKa is equal to 3, that is terminal carboxylic group, pH is greater than pKa, so you get a negative charge. Similarly, for 3.6, you will get negative charge. So add up all together. This is plus 1, this is minus 1, minus 1 and minus 1. You will get minus 2. So P is minus 2. First answer itself is correct. But we will see the others also. Only P minus 2 is shown in first answer. Okay, we will see glycine, serine, arginine, glycine, carboxylic acid. pKa is 8 here and pKa is 3 here for the terminal. Now arginine is having a side chain amino group. The pK value is 12. Similar to this, if you solve, you will get the charge as plus 1. Now R is plus 2, that I have done in the next slide. I will tell it there. Now R is plus 2 charge. Now coming to the cation exchange column question. If the mixture of the above tetrapeptides is separated on a cation exchange column at pH 7. So it is at the same pH they are separating. So the charges won't change that we calculated in question 1. Which tetrapeptide will elute first? So cation exchange column resin is negatively charged. So naturally those which are positively charged, they will bind to the column. Whereas those that are negatively charged, they will get repelled from the column and they will elute first. So here negatively charged is P. So P will elute first. Then comes Q, which is plus 1 charge. And then last to elute will be R, which is plus 2 charge, because the more positive charge peptide will elute last. It will have more tendency to bind to the negatively charged column and it will bind more strongly also. So R will elute in the end. So the answer here is option 1. This is the pK charge of this peptide at pH 7. I have calculated it just the same way as I have done earlier. Here lysine and arginine are having side chain amino groups of pKa 10 and pKa 12. Next one. You have purified an enzyme that has molecular weight of 60 kDa. So the molecular weight of the enzyme is 60 kDa. You run this protein on an SDS page gel and get the following results. Which of the following statement is valid for the quaternary structure of this enzyme? 2 subunits of 30 kDa. If that was the case, you would have got only a single band because 30 kDa will come over here, but since you got 2 bands, that is one at 30 kDa and one at 10 kDa. 1 subunit of 30 kDa and 3 subunits of 10 kDa, that will count up to 60. 3 tens are 30 and 30 plus 30 will be 60. So you will get 60 kDa. 6 subunits of 10, then only one band should have been observed, not 30 kDa band is observed. 3 subunits of 30 kDa and 1 subunit of 10 kDa, that will be more than 60 kDa. So the answer here is option 2. Then next one. What is the V0 Vmax ratio for an enzymatic reaction when concentration of S is equal to 3 km and 9 km respectively? So here we know the Michaelis-Menten equation. V0 by Vmax is equal to concentration of S by km plus concentration of S. So that is equal to 3 km by km plus 3 K3. So that will come as 3 km by 4 km, that will be 0.75. Now next one is substrate is 9 km. So 9 km by km plus 9 km, 9 by 10, that will be 0.9. So 0.75 and 0.9. Answer is option 4. Then next one. Pseudomonas aeruginosa makes blue-green pigment called phycocyanin. To understand the phycocyanin biosynthetic pathway, mutants which cannot make phycocyanin were isolated. 6 such mutants were crossed with each other and the data is shown below. So these are the mutants 1 to 6 and these are 1 to 6 of the mutants. These are crossed with each other. You get some as negative value, some as positive. So negative is phycocyanin negative and positive is phycocyanin positive. Positive means phycocyanin is being produced. Negative means phycocyanin is not produced. Based on this data, can you predict how many genes are responsible for the phycocyanin production? Now this is the case of complementation test. So suppose if there are two genes which are responsible for the production of phycocyanin. One is capital A and capital B. When you cross a mutant of A with a mutant of B, that is small a is the mutant of A and small b is mutant of B gene. So small a into capital B into capital A into small b. These are the gametes and you will get A and B. That is the wild type phenotype. Capital A and B gene have complemented each other and you get wild type phenotype showing that complementation has occurred. But if mutation is in the same gene, that is capital small a. In both the parents, small a gene is mutated whereas capital B is not mutated. You get a mutant phenotype. So no complementation has occurred here. So what you have to do is you have to group together the non-complementation ones as one group because that is the one which is having mutation only in the single gene. So that many genes will be responsible for phycocyanin production. So here if you see, when you cross 1 with 1, this one is negative, 1 is negative and 6 is negative. So there the mutation has happened in the same gene. So they belong to the same group. So 1 and 6 is one group. Then 2, if you see 2 crossed with 2 but with 1, 3 and 4 it has given phycocyanin production. So 2 crossed with 2 is negative. 3 crossed with 3 again negative. 4 crossed with 4 again negative. 5 crossed with 5 again negative. 6 crossed with 1 negative. 6 crossed with 6 negative. We won't take into consideration those values for 6 because we have already taken that into consideration in 1. Because here it is the same gene as 1 and 6. So how many genes are responsible for the phycocyanin production are 1. This is 1 and 6. They belong to one group. So 1, 2, 3, 4 and 5. So 5 genes are responsible for the production of phycocyanin. Then before coming to the next question, I will tell you the endocytic pathway of transferrin receptor or ion transport. So this is the exterior of the cell. The pH is neutral, pH 7. This is the interior of the cell. Now ion will bind to the transferrin. This is the transferrin. Transferrin will bind to its receptor on the outside of the plasma membrane. That is transferrin receptor. It will be It will then recruit the adapter protein and the clathrin to the plasma membrane around where the invagination will take place. Then the a mature pit like this, it will be cleaved off from the plasma membrane forming clathrin coated vesicles by dynamin protein. Then uncoat of the clathrin and the clathrin will be recycled then this will fuse, this will, once it has entered into the inside the cell, it will fuse with the endosome where the pH is 5.55, late endosome where the pH is 5. Remember the pH here, it is 5. Endosome will cause the release of iron. Low pH cause release of iron from the ligand. Ligand remains bound to the receptor. See here, ligand is remaining bound to the transferrin is bound to the receptor. It is not released, only iron is released. Then this transferrin will be recycled to the surface. There it will dissociate from its receptor at pH 7, that is at neutral pH. So this is the endocytic receptor mediated endocytic pathway of transferrin receptor or iron import. Now coming to the question. Iron is taken up by cells via receptor mediated endocytosis utilizing transferrin and transferrin receptor. We saw just now. In a cell line with a mutation in the transferrin receptor that is unable to interact with the transferrin at pH 4 to 6. Now if you see the outside of the cell, I already told you it is neutral pH. But inside the cell, in the endosome, it is pH 5, around 4 to 6. So at that pH, at the pH of 4 to 6, transferrin receptor is unable to interact with the transferrin. That means transferrin will get dissociated from the transferrin receptor at pH 4 to 6, that is inside the endosome. Now, which one of the following steps will be the first affected in this pathway? So, first will be binding of transferrin to iron in plasma. That will not affect because it is at pH 7. It is not at pH 4 to 6. So, binding will take place and it is happening outside. Association of iron bound transferrin with transferrin receptor on the plasma membrane. That is also taking place outside. Association of iron bound transferrin with transferrin receptor on the plasma membrane. That is also taking outside of the cell. pH is 7. So mutation is not going to affect there. Release of iron in the endosome. That also will not hinder because mutation is to the receptor. So release of iron is not going to be affected. But one thing will happen along with iron, the transferrin also will be dissociated because pH is 4 to 6 in the endosome. So transferrin also will get dissociated from the transferrin receptor because the mutation is like that the transferrin receptor is unable to interact with transferrin at pH 4 to 6. So transferrin is going to get dissociated from transferrin receptor and it will be released along with iron. So which is the going to affect in the pathway? Recycling of transferrin to the plasma membrane. That will not happen because there is no transferrin to be recycled back to the plasma membrane. Transferrin already went away along with iron. So answer here is option 4. So this is it. Binding of transferrin to plasma membrane. It takes place in the outside of the cell. It takes place outside of the cell. Then association of iron bound transferrin with transferrin receptor on the plasma membrane, again outside of the cell. Release of iron in the endosome. Here it is pH, low pH, that is around 5, 4 to 6 pH. Transferrin will get dissociated from the transferrin receptor as per the mutation. So iron also will be released, along with that transferrin also will be released. So there is no transferrin, only the receptor will come up to the plasma membrane. So answer here is option 4, that is recycling of transferrin to the plasma membrane. Then next one. In yeast, temperature sensitive mutants of cell cycle regulators CDC2, the key cyclin dependent kinase, CDC13 required for telomeric DNA replication, and CDC13 Rad9 that carries an additional mutation in the DNA damage sensor were isolated. When grown in non-permissive temperature, that is at higher temperatures, for few hours, different phenotypes were observed in these mutants. Choose the option that correctly describes the most likely phenotype for all of these mutants. The CDC2 mutant will arrest in G1S. No, CDC2 is the master regulator. So CDC2 it is required in G1S, G2M phases of the cell cycle. So CDC2 mutant will arrest in G1S, G2 and M. So they will show a different phenotype. See the third option. Then CD13 mutant means cell cycle will be arrested at the G2 phase. So CDC13 in G2 and CDC13 Rad9 will continue to divide for few generations. Now why does it divide? Because due to the mutation in the DNA damage sensor. DNA damage is not being sensed because it is mutated. So it will continue to divide for a few generations. Rad9 is responsible for sensing DNA damage. So that itself is mutated means it will continue to divide for a few generations. So the answer here is option 3. Then next one. The statement below attempt to describe a few characteristics of ALU repeats found in the human genome. ALU elements are a class of short interspersed element signs, combination of all correct statement. Yes, a first statement is correct. Signs are autonomous transposons. They are not autonomous. They require lines for transposition. So they are not autonomous transposons. So that is wrong. ALU repeat originated from the cDNA copies of 7SL RNA. That is also correct. ALU repeat have relatively high AT content. It is not AT content, it is GC content they are rich in. So, D statement also not correct. They are preferentially located in the gene poor G chromosome bands. Now, heterochromatin region, they tend to be AT rich. You all know that. And gene poor also, heterochromatic region, so they stain darkly in G banding. Whereas GC rich region incorporate less Giemsa stain. So here ALU are GC rich, so they will not give G banding pattern. So E is also not correct. So the answer here is C, A and C only. Option 3. Then next one. In C. elegans, PAR proteins segregate at the cell cortex in the zygote to establish cell polarity. This depended on the regulation of the cortical actin cytoskeleton by Rho A. An investigator sought to directly inhibit actin polymerization to analyze the impact of this inhibition on PAR protein. So what you have to see is inhibit actin polymerization. That is the main point of this question. Which of the chemicals would be the most suitable to inhibit actin polymerization is the question that they have asked. Taxol. Taxol, it stabilizes microtubules. Colchicine, it inhibits microtubule polymerization. Latrunculin, that will inhibit actin polymerization. So that is your answer. And LY29402, it inhibits PI3 kinase. So the answer here is option 3. Then next one. A researcher planned to study protein trafficking into the endoplasmic reticulum. For this purpose, they planned different experimental conditions shown below. So each is different experimental conditions. Consider that mRNA that codes for a secreted protein is added in abundant amount. Which experimental control or controls would be the best to confirm the polypeptide entry into the endoplasmic reticulum? Okay. Now we will see the first control. So proteins are fed into endoplasmic reticulum during translation if they have amino acid sequence called signal peptide. So signal peptide is required for the proteins to enter into the endoplasmic reticulum during translation. Now we will consider that all that signal peptides, everything is ready. The first one, the cytosol is mixed with mRNA that codes for a secreted protein. So you take the cytosol, mix it with mRNA in a test tube. For the secreted protein, mRNA is added in abundant amount. Then this is followed by Western blotting with antibodies against secreted protein. So after that, translation happen, it will enter the endoplasmic reticulum. Oh, they have not added endoplasmic reticulum here. So this will just prove that the mRNA codes for the secreted protein. So this can be used as one control. Whatever mRNA you are using, it codes for secreted protein is confirmed by this particular experiment. So A can be used as one control. Then next one. The cytosol is mixed with mRNA that codes for a secreted protein and rough microsomes. Rough microsomes means small endoplasmic reticulum particles carrying ribosome, followed by Western blotting with antibodies against secreted protein. Here the protein formed via translation of mRNA using the cytosolic component, and it will also enter the microsomes. From there the protein is extracted and subjected to Western blotting with antibodies against the secreted protein. You will get the result that it has entered the ribosome microsome and secreted proteins are produced. So that also can be used as a control. Then next one, the cytosol is mixed with mRNA that codes for a secreted protein and rough microsomes followed by protease treatment. Subsequently, Western blotting with the antibodies against the secreted protein is done. Here also it will enter the microsome. Then it also shows that even after protease treatment, for the secreted protein could be done, showing that the protein enters the microsome. That is why it did not get cleaved by the protease. If it was cleaved by the protease, then Western blotting would not have given result. But since it entered the rough microsomes, it was protected from protease cleaving, and Western blot gave the results. So A, B, C can be used as, this is also a very best control for the experiment. So ABC can be the best control, as it will show that mRNA producing the protein as in A, the protein entering the microsome as is the case of B, and a protein entering into the microsome again confirmed by protease treatment. So A, B and C can be used as control for this particular experiment. So the answer here is option 4. Then next one. Following statements are made about mRNA splicing. Involvement of cis-acting branch point. Now before that, I will just brief you with mRNA splicing. This is exon 1, this is exon 2, this is 5' splice site, this is 3' splice site. So what happens here is a cut will occur here, and the intron will be forming a the G of intron will attach with A at the branch point, and exon 1 will be released. Now exon 2, here also another cut will take place, that is at 3' splice site, and the intron will be released as a lariat, and exon 1 and exon 2 will be joined together. This is what is happening in mRNA splicing. So involvement of cis-acting branch point or branch point sequence present near 3' end of each exon is essential for splicing. It is not essential. The branch point site is on the intron. See this figure over here, it is in the intron. So first statement itself is not correct. They have asked for all correct statements. So for A is not correct. So 3 option is also not correct. 3 option is also not correct. Okay, we will see the others. In the first step of splicing reaction, 2' OH of the conserved U at the branch site act as a nucleophile to attack the phosphoryl group of the conserved G in the splice site. It is not U. It is not U here. It should be A. See here in the figure. It is A here. A will have a nucleophilic attack on the G. It is not U. So B is also not correct. So again 1 and 4 have been cancelled, and your answer is option 2. Now the newly liberated intron adapts the shape of a lariat due to joining of the 5' end of the intron to the branch point. That is correct. During the splicing process, there is no net gain in the number of chemical bonds, because there are 2 trans-esterification reactions. First is the cleavage that take place at the 5' splice site, and a lariat is formed by the attachment of the 5' end of the intron to the branch point. Second cleavage take place at the 3' splice site, and 2 exons are spliced together, and in this trans-esterification reaction. So these are the 2 trans-esterification reactions. That is, first cutting of the 5' splice site, joining it to the branch point, that is first trans-esterification reaction. Then 3' splice site is cut, both the exons are joined, the intron is released as a lariat, that is the second trans-esterification reaction. So there is no net gain in the number of chemical bonds. So D is also correct. Then E. PRP22, a dead box helicase, is required for stripping the spliced mRNA from the spliceosome. That is also correct. So answer here is option 2, that is C, D and E.